Hello, everyone. Good morning, and thanks for taking your time to attend this important webinar. My name is Trisha Wood. I'm a part of the North American Ruminant technical team here at Lallemand Animal Nutrition. I'm delighted to host this live webinar session with all of you today. Today, I'm hosting two experts. Dr. Frederique holds a PhD in microbiology, and she is head within Lallemand Animal Nutrition for the ruminant tech research with high expertise on rumen microbiota. She is based at the ENRA in France. Dr. Emiliano holds a PhD in animal science, and he's from Cornell University. He's currently working as a consultant for Rumen and NDS professional CNCPS-based platform, and he is also an assistant professor at the University of Padova. He's based out of Italy. Thank you so much for being here and for both of you accepting this challenge. It's always great to have you with us. This session is the second one of a series of 2 episodes where we will discuss the importance of fiber digestion and the rumen environment from rumen microbes to diet formulation. Before starting, please be informed that this webinar is going to go about 50 minutes with a 15-minute question and answer session. We invite you to place any of your comments or questions in the small square that you have in your control box menu. These are going to be anonymous, and it's usually located at the bottom area of your control box. At the end of the presentation, I will field these questions to the relevant speakers. Before turning the presentation over to our 2 great speakers, let's go over a summary of what we discussed in the first session. First, the rumen ecosystem is engineered to be centered around degradation of a plant fiber by a complex microbial community. The fibrolytic microbiota is very active, but also quite sensitive to its environment. They may have negative impacts on fiber digestibility, feed conversion, and finally production. Dynamic models like CNCPS are able to integrate this rumen dynamic, and these models are very sensitive to characteristic inputs. The most important nutrient input is NDF, and changes in its digestibility values will result in different ME and MP allowable milk. Modifying NDF digestion will allow nutritionists to reformulate diets and adjust the composition according to actual economics and farm manager objectives. Income over feed costs remain the most important affected value. Using a rumen modifier, such as Levicel SC, helps promote or stabilize the microbiota, and we now have expertise on its benefits in a variety of dietary and stress situations. Thanks to improved fiber digestibility, ruminants extract more fiber from the diet, which is particularly relevant in today's agricultural systems. Frederique, I invite you to open your mic and please explain to us how these microbes are affected by the rumen environment.
Thank you, Tricia, for this kind introduction, and hello to everyone. So as Tricia mentioned, I'm Frédérique Chauchera-Durand. I'm a microbiologist working for Lallemand Animal Nutrition. And today I'm going to show you how crucial the rumen microbiota is for fiber digestibility and how it can be affected by challenges that producing ruminants encounter in their day-to-day life, with a particular focus on ruminal acidosis. First of all, it is obvious that fiber is and remains a major component in all ruminant diets worldwide. The main digestible part is constituted by plant cell wall polysaccharides with a diversity of polymers which are more or less accessible, such as pectins, hemicelluloses, and cellulose. Thanks to the unique ability of ruminants to convert this plant biomass into energy and finally edible products, these animals are not in competition with humans for their nutrition. And they participate as well to land architecture, so they are really essential in sustainable agricultural systems nowadays. Fiber degradation cannot be ensured by any host enzyme, and ruminants have developed a strong symbiosis with a complex and specialized microbial community called microbiota, inhabiting the rumen compartment, which is in fact the main site for fiber degradation. The 3 main groups that are found in the rumen are bacteria, ciliate protozoa, and fungi, and we can also find archaea and viruses as well. These microbial communities differ in abundance and in size, but most of them are considered as strictly anaerobic as they cannot live in the presence of oxygen. The more specialized fibrolytic microbiota belongs to these three microbial groups. Among the bacterial community, fibrolytic populations represent around 5% of the total and are represented by very specialized species. In the ciliate protozoal community, these are mainly antodinomorphid species, which are really recognized as fibrolytic. Finally, in the fungal community, all isolated species have been proven as exhibiting high enzymatic activities on cellulose and hemicellulose. The activity of fibrolytic microbiota results in the generation of a trophic chain that is described here, which starts actually by fiber degradation by the fibrolytic communities. So each step of this trophic chain is important. First, these fibrolytic microbiota are able to break down the plant cell wall polysaccharides into soluble compounds, sugars and oligosaccharides, which are in turn reutilized by other species to produce fermentation intermediates, with finally generation of end products of fermentation, which are mainly gases under the form of CO2 and methane, and volatile fatty acids, mainly acetate, propionate, and butyrate, which represent around 70% of the energy needs of the animal. Keeping an active fibrolytic microbiota at the start of this process is thus essential to ensure a good rumen function. In its life, a ruminant undergoes a huge anatomical, physiological, nutritional, and even psychological challenges. From calving and calf's birth, weaning, growth of the pre-productive stage, to gestation, parturition, and further production, lactation or fattening. These challenges may be even worsened by heat stress conditions or changes in herd management practices, for example. Since the last decades, a lot of research has been devoted on investigating the impact of some of these life cycle steps on rumen function and health, and this research has revealed that during all these periods, the rumen microbiota and the rumen epithelial wall as well can also encounter many challenges as their environment is submitted to numerous variations, such as changes in pH and oxygen levels, changes in nutrient sources, or adhesion sites. Fibrolytic microbiota is particularly sensitive to changing conditions in the rumen environment, and this can impact fiber degradation, of course, animal efficiency, and well-being. Today, I will focus on acidosis, which is a well-known disorder in high-producing ruminants fed high-concentrate diets. Acidosis originates from a microbial imbalance, which occurs in the rumen when rapidly fermentable carbohydrates are fed. These substrates are quickly degraded and fermented by rumen microbiota, which leads to the production of VFA, initiating a decrease in rumen pH. This decrease in rumen pH favors rapid growth of lactate-producing bacteria, such as Streptococcus bovis and others. Lactic acid is known to be 10 times stronger than VFAs, so even low amounts of these acids are able to decrease rumen pH. Some beneficial species, such as Megasphaera alsdeni, are able to utilize lactic acid and thus limit acidification of rumen contents. The balance between lactate-producing and lactate-utilizing bacteria is thus key in maintaining a physiological pH, and when the balance is disrupted, we talk about subacute ruminal acidosis, or SARA, or even to acute acidosis when the pH is below 5.5. Beyond pH and microbial imbalance, under SARA, there are many events that may be at the origin of severe problems, such as rumen wall damage and transfer to the bloodstream of toxins and inflammatory molecules, such as lipopolysaccharide or histamines, that can trigger a more systemic inflammatory response. With most diets, SARA is a threat for dairy and feedlot cattle worldwide. It is still quite difficult to accurately diagnose SARA, as it triggers various changes that are sometimes rather diffuse to identify. Rumen pH measurements can be a very good way to identify SARA, and since the last 10 years, the development of reticulorumen pH sensors that can be called rumen pH boluses are applicable on farm to characterize SARA risk. These sensors allow to monitor pH kinetics and thus determine mean daily pH, minimum or maximum pH, and define SARA threshold. It is important that, to remind that these sensors are located generally in the reticulum, and pH values recorded with the bolus are always higher in this compartment compared to the rumen compartment, with a difference which has been established at around 0.2 to 0.3 units. So here on the graph on the left, you can see an example where pH has been measured with boluses or with probes inside the rumen with solid and liquid phase of the rumen contents. So here you can really very easily see the difference that exists between the measurement with the bolus and the measurement with probes in rumen samples. This difference has been really to take into account when defining a SARA threshold. Recently in Lallemand, we have worked on the development of new indicators for reticulorumen pH, which are interesting in order to better characterize the individual variability between animals. And here we consider pH amplitude, as you can see here, as a relevant parameter to be taken into account. These relative pH parameters, on top of the classical pH parameters, can be very interesting to better describe and understand the sensitivity of animals to SARA. A recent multicenter study has been recently published by Golder and co-workers from Australia. And in this study, they have evaluated the acidosis risk in dairy cows from herds in the US, Canada, and Australia, with a quite large range of diets regarding non-fiber carbohydrates, NFC, or neutral detergent fiber, NDF. The analysis was conducted on 261 early lactation Holstein cows from 32 herds, taking into account a combination of individual rumen parameters such as rumen pH, ammonia, lactate, or VFA concentrations. The agglomeration of these variables into a discriminant analysis model led to the definition of 3 risk groups: high, medium, or low. And you can see from these pie charts that in particular in Australia and in US, in California, for instance, the proportion of cows who were actually classified at high or medium SARA risk was actually higher than 60%. So it was a huge proportion. In this same study, rumen microbiota was monitored by sequencing the DNA extracted from the whole microbial communities in the rumen. And here you have bacteria composition and diversity according to the acidosis risk group. Very briefly, because the paper is quite rich, what we can see here is that proportion of the main bacterial taxonomic groups, Firmicutes, Bacteroidetes, and Actinobacteria, were really different in the high risk group compared to the other groups, with more Firmicutes and more Actinobacteria, and while less Bacteroidetes in the high risk group compared to the medium and low risk, which was indicative of a more unstable rumen ecosystem, which was also highlighted by the fermentation profiles. The bacterial diversity, which is shown on the left here, was clearly decreased in the high risk group, as you can see on these box plots, and this was really indicative of unfavorable conditions for an optimal rumen function. So generally, decreased diversity means less favorable ruminal conditions for ruminant functions. With a metatranscriptomic approach, allowing to assess really active bacterial populations, the effect of SARA challenge was studied here in this example in beef cattle. Here you can see that the beta diversity, which allows to compare global rumen microbial profiles between the two groups of animals, so challenged were in blue and controls were in yellow, was clearly different. You had different clusters with the different microbial profiles. The histogram below shows, in fact, the discriminant analysis where some particular taxa could be identified as markers of either the control in blue or the control in yellow, or the challenge in blue situation. For instance, if we take the first one, Fibrobacter succinogenes, this is a highly fibrolytic bacterial species. This species was strongly positively associated with the control diet and not at all with the challenge diet. On the heat map presented on the right side, the gene expression profile of the microbiota were compared in the two groups of animals. And in fact, the several genes of the rumen microbiota were found overexpressed in the challenge group. Within these highly expressed genes, there were genes involved in biofilm formation. The most expressed genes are highlighted here in the red color. It is important to notice that biofilm formation is a strategy employed by pathogenic bacteria to maintain and resist to antimicrobial agents. So these data together suggest that animals under SARA would be more at risk to harbor antibiotic-resistant pathogens. If we look more in detail in fibrolytic populations, a lot of studies now report a negative effect of high starch diet and low rumen pH on these populations. Here, you see an example from an in vitro study where SARA was induced by a change in forage to concentrate ratio. The Rizitec model is able to simulate rumen fermentations. And here you can see on this graph here below that indeed it responded quite well to the SARA challenge with a strong decrease in pH. Looking at microbiota, total bacterial abundance was not affected, as you can see here, whereas protozoa and fungi were very strongly decreased in the SARA group. Within the bacteria, however, some specific fibrolytic bacteria were deeply negatively affected, as you can see on the graph here on the right side. So overall, these in vitro results indicate a high sensitivity of the fibrolytic communities to low pH. In vivo, the impact of SARA has been also clearly demonstrated in numerous studies. Here, I will show only two examples. The first one shows a very marked decrease in relative abundance of Fibrobacter genus, which is the fibrolytic bacterial genus I just quoted before, and of Polyplastron, a ciliate protozoa genus, which is very active in plant cell wall degradation. The reduction of abundance of these different genus was more and more important according to the level of concentrate in the diet. This was also correlated to a decrease in rumen pH and of the acetate and propionate ratio. The second example on the right results from a metagenomic study in dairy cows where a gene catalog of the whole rumen microbiota was established, indicating the potential of microbial activity. Increasing the level of starch from 20 to 33% had a negative impact on the proportion of these genes encoding for fibrolytic activities and for microbial proteins involved in bacterial attachment to cellulose. So a strong impact also on microbial fibrolytic potential. It must be reminded that ruminal acidosis may occur in other dietary situations than with high starch diet. Indeed, spring pasture, for instance, contain high levels of fermentable carbohydrates and low cellulose contents, which may put the animal at risk for SARA. Studies conducted, for instance, in Ireland showed that an important proportion of cows grazing perennial ryegrass pasture were at risk by being a substantial amount of time with a reticulum pH below 6. So in fact, this is mainly due to fructan content of grasses, which can vary greatly depending on the type of grass, time of year, and growing conditions. The content has been reported to vary from 3 to 50%. So the fructan content of grasses has been shown to be very favorable to fast growth of specific bacteria, but not at all favorable to fibrolytic species, unfortunately. In an in vitro study conducted at USDA by Paul Weimer and Maribeth Hall, the maximum growth rate of isolated rumen bacterial strains was measured using different types of substrates, in particular here phlein, which was the fructan found in orchard grass, and sugar-derivated fructose and sucrose. You can see on this table here that the fermentation of fructans and their derivatives was really very efficiently achieved by saccharolytic species like Prevotella, Selenomonas, Streptococcus, or even Succini vibrio, as shown in bold with the very high growth rates. In contrary, the fibrolytic species Fibrobacter succinogenes and Rumenococcus albus or Rumenococcus flavefacens were totally unable to ferment phlein, fructose, or sucrose. So the fermentation of these sugars by saccharolytic bacteria led to the production of high VFA and high lactate concentrations in the rumen, which of course increase the risk for SARA. In addition, the production of capsular exopolysaccharides from Streptococcus bovis could also participate to the generation of frothy bloat, so secondary problems which can be originated through acidosis. So we have seen that SARA occurs in many situations in cattle, and there is a need to secure and preserve fibrolytic communities. So I will take an example of the use of a rumen modifier, namely the rumen-specific yeast Levusalaceae, which is a live strain of yeast of Saccharomyces cerevisiae, for which we have strong proofs of efficacy on rumen function improvement, in particular on the promotion of fibrolytic microbiota through the control of rumen pH. So this live strain, which is named CNCM I1077, exerts different modes of action. Nutrient supply at the microhabitat level and nutritional interactions with the rumen microbiota are able to promote growth and persistence of the fibrolytic populations. The yeast cells are able to scavenge oxygen traces in rumen contents, which creates more favorable anaerobic conditions for adhesion and fiber colonization process. This live yeast strain can interact with lactate producers and utilizers, which leads to the control of microbial balance, reduction of lactate accumulation, and stabilization of rumen pH. All in all these beneficial effects help to influence positively fibrolytic microbial communities. The live yeast strains limits lactate-producing bacterial growth, as we can show here, thus maintaining low levels of lactate and higher pH. Secondly, the live yeast is able to stimulate the detoxifying action of lactate-utilizing bacteria, further decreasing the level of lactate in the rumen. Studies in dairy cows equipped with reticulorumen pH boluses have confirmed that Levusalaceae treated cows show a clear reduction in the time spent under a certain pH threshold, here 5.6, as you can see on this table. And you can see the pH kinetics here that are really better, have a better shape with the Levusalaceae treatment. By acting on the microbiota balance, Levusalaceae has then the capacity to deeply alter the microbial ecosystem in a very beneficial way. There are today numerous in vivo trials that confirm that CNCM I1077 is able to stabilize rumen pH in case of SARA risk. And this is a summary of some recent trials that have been undertaken in Lallemand animal nutrition. Interestingly, more stressful were the conditions, so where you have the lowest rumen pH, higher was the effect of Levusalaceae on stabilization. Besides this improvement of mean rumen pH, the live strain was able to limit pH drops after thinning, and by this way, stabilizing rumen environment. In particular, fibrolytic activity can be maintained in the presence of LC. And we illustrate that by this in vivo study, where we have measured several enzymatic activities in the solid phase of the rumen contents in sheep fed a high acidotic diet, which was a high level of barley. As you can see, the high starch diet in yellow induced a very strong decrease in enzymatic activities in the control sheep, compared with the same sheep fed with the high forage diet in light green. When the animals were fed with the LC in bright green, you see that the supplementation allowed to maintain a higher level of these enzymatic activities, which was, of course, linked to higher pH that was measured in parallel. So finally, I would like to take home a few messages. First, that producing ruminants are often at risk for rumen dysbiosis, in particular at the rumen level for SARA. SARA is a rumen disorder which has a negative consequence beyond the rumen, so it can go really beyond this compartment, and it can be triggered by a variety of diets, not only high concentrate diets, and can be also amplified by additional factors like heat stress or poor management. Rumen microbiota, and particularly fibrolytic communities, are very sensitive to acidosis conditions, which leads to decrease in fiber digestibility and overall poor performance and health deterioration, as fiber digestibility, as you know, is a key process in ruminant physiology. So to prevent SARA, one strategy would be to use a live yeast additive such as LC, which was really shown to stabilize rumen pH and rumen microbiota, with final benefits on fiber digestibility, feed efficiency, and animal performance. So I thank you for your attention, and I will take any question at the end of this webinar. Thanks a lot. Bye-bye.
Thank you, Dr. Frederic, for this timely and interesting information. It's clear that rumen microbes are sensitive to the rumen environment. I will now give the mic to Emiliano. Thanks, Dr. Emiliano. Could you please help us understand how this rumen pH is impacting NDS outcomes?
Sorry, I have two screens. I'm trying to. Okay, can you see my screen?
Yes, we can see the screen.
Okay. Well, thank you for the introduction, and thank you to Lallemand for the opportunity. So first of all, I would like to say that you know, the objective today of this presentation is trying to understand how, you know, all the science findings regarding rumen pH and SARA, obviously, are implemented in the platform or in a model like the CNCPS and NDS. And you'll realize right away that you know, we are really far actually from being able to, you know, dealing with all the complexity that Frederic has been you know, telling us about rumen pH in you know, in ruminants. So obviously, just to re-emphasize the concept, you know, we all know that rumen pH is basically the balance, you know, between production of acids and removal of these acids. So obviously, anything that will affect any of these two, production or removal and absorption, will basically result in an imbalance of the rumen pH. And obviously, acidosis is what is relevant from a production point of view. So what does the CNCPS do to deal with this risk with the rumen pH? So there is a rumen pH prediction in CNCPS, and unfortunately, as today, it deals only with physically effective NDF. So as you can see from the graph, this is a simple relationship between physical effective NDF and rumen pH. So not only is simple, but you can also see that you know, it has a plateau at about 23 24 physical effective NDF, and then there is a slight decrease, and to reach a pH of 6, we have to basically go down to a physical effective NDF of 15%, 14%, which is not very common in commercial farms. But we also know that the rumen pH of 6 can be also very frequent in higher producing cows. So what does the CNCPS do then with these values, with these numbers of rumen pH and physical effective NDF? So there is an adjustment related to the non-fiber carbohydrates bacteria, only for physical effective NDF lower than 20%. Okay, so there is, as you can see from this graph, in this case, the relationship is between PE-NDF and NFC bugs. So when the PE-NDF goes below 20%, there is a reduction of the bugs produced in the rumen. And as you can see, there could be even a 200 grams reduction of microbial production due to a physical effective NDF. So if we decrease a PE-NDF from 20% to 16%, for instance, we have a reduction of about 250 grams of microbial protein. That corresponds to actually 2 kg of metabolizable protein allowable milk. So these sometimes it's kind of underestimated by us, by MDS and CNCPS users, but there is an effect on microbial protein production. So that is not related directly to pH, but to physical effective NDF. So this should be an incentive for us to actually measure PE-NDF as often as we can in the farm at the TMR, in the TMR level in the field. So, for example, in NDS we have a tool that allows the users to adjust the physical effective NDF of the whole TMR. So obviously, the more precise and accurate we have this value, the more reliable will be the prediction of MP allowable milk. So what happens for the FC bacteria, which actually they should be the ones that are more affected, it is only a very slight reduction. So we're talking only about a few grams of reduction of bacteria produced, and it comes mainly from a reduction of the KD of the B3 fraction, so of the potentially digestible NDF. And that reduction of the KD is actually related. It is dependent also on the initial KD, so on the baseline KD. As you can see, this is the reduction of the KD according to the rumen pH, and the slope is actually higher for higher quality forages. So the higher the KD of our forage, the more important is the reduction of that KD due to a reduction of rumen pH. However, if we go back to the slides, we have also to remember that to have a pH lower than 6, we have to go to a physical effective NDF that is actually lower than 14–15%. So as you can see, it's not a very important reduction in terms of microbial production coming from the FC fibrolytic bacteria. But if you are interested in understanding how important this reduction is, NDS also has on display actually the relative yield of the FC bacteria that is here to understand how much those bacteria actually reduce because of the physical effective NDF and because of pH. Now, the previous presentation has highlighted how the SARA incidence and severity is a very complex issue coming from different factors. Okay, and unfortunately, the CNCPS as today deals only with the physical effective NDF, which it is an important tool, but it deals mainly with the physical effectiveness of a TMR diet. So there are many factors that are excluded from this prediction. So what are those other factors? These are only some of them. So they could be amount and fermentability of starch. It could be the feed intake level. It could be also the nutritional management. And we need to actually remember how our goal is not only to have a good fermentation, but it is to have a consistent fermentation across the 24 hours. So anything in the nutritional management that kind of interferes with this consistent fermentation will also alter the rumen pH and increase the risk of subacute acidosis. So because of all these factors, NDS has tried to implement an alternative model for the prediction of rumen pH. So it does use a meta-analytic approach, which means we have collected various numbers, various data from different research trials coming from both dairy cows and beef cattle. And we have built a complex algorithm predicting rumen pH. So I will just go over the main adjustments, the main equations of the pH prediction. So it is based on two main steps. So the first step basically sets a baseline that is estimated by the intake of forages and concentrates. So in particular, the concentrates intake as a percentage of body weight will be the determinant, the main determinant of the baseline pH. Then this baseline pH is adjusted based on various factors. So NDF, PNDF and particle density, rumen degradable starch, and the possible presence of buffers in the diet. So how these adjustments are implemented quickly. So there is an adjustment for an NDF that goes below 37%. So if the total NDF is 37% or higher, there is no adjustment. Otherwise, as you can see, there is a reduction, an adjustment to that baseline pH that I mentioned before. Then there is also another adjustment based on particle size. So if the diet has a particle mean size of 3 millimeters, you'll see that there is also an adjustment, a negative adjustment of that baseline pH, but above actually that 3 millimeters, there is a positive adjustment. Similar things happen with the ruminal degradable starch. So the center is set at about 17%. And obviously here I'm dealing with ruminal degradable starch, which is predicted by the CNCPS itself. So as you can see, there is a negative adjustment for values that go above that 17%, and a positive adjustment for values that go below that 17%. Then lastly, as I mentioned before, there is an adjustment that is dealing with the presence of buffers. And as you can see, the adjustment is not linear, but it basically depends on the initial pH. So the lower the pH, the larger the slope. So meaning the more important the adjustment will be. Okay, so obviously, if I have a higher pH of the baseline pH, the presence of buffers, which is what the science has demonstrated, will not really affect the rumen pH. So going back to NDS, as we well know, we don't only need to deal with the average pH, which is what we usually do, but we also need to deal with the range of pH across the 24 hours. So before I mentioned that the fermentation or the consistency of the fermentation across the 24 hours is actually what matters in terms of incidence or risk of acidosis. So what NDS does is not only predicting the average rumen pH, but also the range of rumen pH, so minimum rumen pH, maximum rumen pH, and the number of hours that this pH goes below either 5.8 or 5.6. And these numbers were chosen because, in fact, there are various papers showing how these thresholds should be used, in terms of associating pH to the presence or the risk of acidosis or subacute acidosis. So these number of hours that pH is estimated to go below 5.8 is actually used in NDS to define also an acidosis index, which again is defined, is calculated based on those number of hours below pH 5.8, and then adjusted by the dry matter intake of the individual group of cows that we're dealing with. Based on that, we'll have an acidosis index that will suggest that there might be a risk or no risk of acidosis in the group of animals. So again, these are tools kind of have the objective of assisting the nutritionist, the technician, to obviously understand if there is a risk. There is also another tool in NDS that deals with simulating the effect that a rumen pH can have on the digestibility of NDF. So if we go into feeds utilization, we can actually see how the rumen pH that is predicted by NDS can quantify actually what is the expected decrease in terms of PD NDF digestion. So in this case, the pH, as you can see, is not low, so it's 6.16. So there is a very, very small reduction of 33 grams per day of PD NDF digestion. But there is also obviously associated to this a reduction of only 22 grams in terms of bacterial yield. And obviously these estimations come from the rumen pH predicted by NDS. Another tool that we have in NDS is also that we simulate basically how the KD will be reduced or should be adjusted because of that pH reduction. So as you can see here, we have an example of one feed of a corn silage that has a B3 KD of 669, but it actually should be reduced. We believe it should be reduced to 6.16 because of the pH that we have right now in the rumen. So this is true only for forages, and we'll actually leave the users to freely adjust the KD or not. So the KD is maintained at 6.69, but then the user is free to choose if to reduce that KD or not. Then last, we have a more recent tool in NDS, which is not really, it was not really built, to assess rumen pH or to work with acidosis, but it can actually give us some help to synchronize the rumen environment a little bit better in terms of degradation between carbohydrates and protein sources. That I invite you actually to look at it if you haven't done it yet. So our take-home messages is that the CNCPS prediction is there. It is very simple and based only on physical effective NDF, because basically most of you all know that most of the literature present is relating pH to physical effective NDF. So when it was built, most data were coming from those studies. And there is, however, an effect on metabolizable protein coming from microbial production. So what we've been trying to do in the last few years is trying to fill this gap that the CNCPS has in terms of pH prediction with a more complex, a little bit and quasi-mechanistic pH prediction using different factors. So, but in the end, the results are an alternative pH prediction, but also the range of pH prediction across the 24 hours that will give you an index of acidosis, basically the risk associated with this range of pH across the day. So besides this, NDS provides also simulation for the PD NDF KD reduction if we believe there should be a reduction, and that obviously the user again is free to implement that KD, the suggested KD or not. So in the end, as I mentioned before, it's not only about average rumen pH across the 24 hours, but it's all the fermentation that should be working well across the 24 hours. So it's not only about amount of starch, it's not only about feeding effective NDF, but also, as Frederick mentioned before, it's also about nutritional management. So avoiding overcrowding, trying to push up the feed, giving the TMR more times per day. So all these factors will actually help the ruminant to have a more consistent fermentation across the day. So if we don't have time today to answer all the questions, please feel free to email me using this email address. But I will try to answer any question now. Thank you.
Thank you, Dr. Emiliano, and thanks again, Dr. Frederick, for your presentations. Lallemand is committed to bringing fundamental research to the field. This webinar is a good example of how Lallemand is bringing services and solutions to address animal challenges. Significant progress has been made in the model refinements over the past several years through the inclusion of biological and dynamic pathways for ruminant digestion. These nonlinear refined models provide a path for innovative formulation systems, which offers opportunities to fine-tune the prediction of nutritional values in diets, including potential submodels with rumen modifiers. Thank you all for your highly valuable participation